Alrighty, so I'm going to start introducing everybody. First, I'd like to say when we get to the Q&A session, please raise your hand and then I'll be able to select you to be able to talk. And then you'll be able to unmute yourself and ask your question. Be sure to unmute yourself afterwards. I appreciate it. So I'm going to officially start. Welcome everybody. Yeah, I'm really super excited to be here with you all with this edition of the ASBNB Lipid Research Division Seminar Series. And so my name is Michael Hanna, and as of January, I am an assistant professor in the Department of Biochemistry and Molecular Biology at Penn State University here in Happy Valley. And really, it's an honor to be hosting this seminar with our two outstanding speakers today. And so the theme really is new insights into the mechanisms and functions of non-vesicular lipid traffic within cells. And at least part of the focus today is going to be on this really fascinating protein called BLTP1, which is formerly known as KIAA 1109. It's also known as a cold-sensitive fermentation CSF1 in yeast and lipid depleted gene 3, LPD-3 in worms, or tweak in flies, named after, of course, the beloved South Park character. Now, the work by today's two speakers have really characterized BLTP1 like never before. And I think more broadly, maybe more importantly, their work has really revealed, I think, spectacular mechanistic details about the function of these bridge-like lipid transfer proteins, which BLTP1 is a member. So these BLTP1s are ideally suited for the bulk transfer of lipids or membrane lipids between organelles, which is still relatively a new idea in cell biology, and there's vast potential for new insights, which is what you're going to be getting today with today's seminar. So with that, it's my pleasure to introduce our first speaker, Dr. Sarah Clark. So Sarah earned her PhD at Oregon State University and did her postdoc at the Ballum Institute at Oregon Health and Science University. She opened her lab in 2023 at Oregon State University in the Department of Biochemistry and Biophysics. And we're all very much looking forward to your talk, Sarah. So with that, the floor is yours.
Excellent. Awesome. Thank you very much, Michael, for giving me the opportunity here to tell you about our work on BLTPs. So the focus of my lab is trying to understand how BLTPs, or bridge-like lipid transport proteins, work by studying their structure. At our core, my lab is a structural biology lab. So we study large protein complexes, typically membrane protein complexes, in order to understand their mechanism and how they're functioning inside cells. So today I'll be telling you about our work on the BLTP1 complex and what we've learned about the mechanism of lipid transport by looking at the cryo-EM structure. So first, just a little bit of background on the importance of lipid transport within a cell and on BLTPs and their role in lipid transport. So here in this image of a cell, this sort of classic cartoon image of a cell, pretty much every one of the organelles that you see is surrounded by a lipid membrane. We have the endoplasmic reticulum shown here, mitochondria, Golgi apparatus, et cetera, the plasma membrane itself. And all of these are surrounded by a membrane, which has a very specific composition. And also these membranes are highly dynamic. So the membranes within a cell are constantly undergoing various processes that are really important for the function of a cell, including processes like phagocytosis, where the cell engulfs things within its environment, exocytosis, where it releases things into the extracellular environment, organelle biogenesis, membrane homeostasis, temperature adaptation. All of these processes are really fundamental to the life of a cell, and all of these processes require an influx of new lipids in order to make them happen. Most of these lipids, the vast majority of cellular lipids, are made in the endoplasmic reticulum, this purple structure here. And then they have to be trafficked everywhere else in the cell by crossing the aqueous cytosol in order to get to where they need to go. And one of the main ways that this happens is through the action of lipid transport proteins. So lipid transport proteins, there are dozens of them within a cell, and a lot of them operate via this shuttle mechanism that's shown in the cartoon here, where the lipid transport protein sort of grabs one lipid, it's anchored to a membrane on one side, and grabs one lipid, moves it to the next membrane, another lipid moves it to the next membrane, and in this way shuttles between the two, typically moving around 1 to 4 lipids at a time. And in contrast to this, bridge-like lipid transport proteins, which are more recently discovered, and were only discovered somewhere around 6, 7 years ago, they move lipids through a different mechanism. So they also exist at these membrane contact sites, regions of the cell where two membranes are in very close proximity. And at these membrane contact sites, like Michael mentioned, they're thought to be responsible for bulk lipid transport. So they form a physical bridge between these two membranes, and where one end of the, if you visualize it like a tube, is connecting to the place where the lipids are made, and the other end is connecting to where the lipids need to go. And in that way, they're shuttling lipids from one place to another in a bulk fashion. So for that reason, they've been described as things like a hydrophobic conduit, a lipid fire hose, a lipid superhighway, things like that. There are 5 BLTP family members, which are numbered 1 through 5 here, but they're more commonly in the BLTP field, known by other names, such as VPS13 for BLTP5. And these are the predicted structures made by AlphaFold, shown on the right here. And you can see that they have sort of a base structural similarity. They all have this beta strand backbone, shown in green here. Some of them, like BLTP1 and BLTP2, have an N-terminal transmembrane helix, whereas the other 3 do not. And none of them have a C-terminal transmembrane helix to anchor them to the adapter membrane, but most of them have amphipathic patches or hydrophobic patches at their C-terminus that are thought to enable interaction with the acceptor membrane. And they differ in various ways. Some of them have extra domains, and they're also, obviously, some are much longer than the other, with BLTP1 being the longest and the largest of all the BLTP proteins. So we have been studying BLTP1, which is also known as LPD-3 in C. elegans, and I'll be referring to it primarily as LPD-3. And we're interested in understanding how it works. So we want to know the function of this protein, primarily, like I mentioned, by studying its structure. So we want to solve the cryo-EM structure of BLTP1 in order to figure out the mechanism, how lipids are physically being moved from one membrane to another. And our approach to this project was a bit unconventional. Because we knew from other people that expressing BLTP1 recombinantly in any cell type was very difficult. It's a large protein. It's 450 kilodaltons in C. elegans and about 550 in human. And it was unknown at the time what auxiliary proteins associate with BLTP1, which also is an important factor to consider when in macromolecular assemblies. So since it was not easy to recombinantly express, instead, we decided to purify the native protein complex from C. elegans. So in order to do this, we generated a transgenic worm line. And the reason we chose C. elegans is because they're easy to genetically modify. So we could use CRISPR-Cas to knock in a tag at the terminus of BLTP1. And also because my lab had previously developed methods to purify native protein complexes from C. elegans in order to study their structure and function. So we applied these methods that we had developed to the isolation of C. elegans LPD-3 And it worked beautifully, actually. So what we did exactly is we used CRISPR-Cas to create a transgenic worm line wherein a fluorophore and a 3X flag tag were at the C terminus of BLTP1. And then we did a bunch of validation assays to make sure that the protein is localized correctly and to make sure that it's still functional. So when BLTP1 is not functional in C. elegans, the worms are very cold sensitive, and that's one test among a few. And we determined that this particular worm line, BLTP1, did appear to be functional. They were behaving like wild type, and so we could move forward. So the next thing we did was grow lots and lots and lots of worms. And that's because for downstream structural and analytical studies, we needed a lot of protein in order to accomplish these studies. And native protein is just expressed in such low abundance compared to a recombinantly expressed protein. So we typically grew our worms in liquid culture in sort of a similar way that one would grow E. coli, and then harvested them in these little Dippin' Dot type things that were then ground up, solubilized, and then we use sort of conventional biochemical techniques to extract the protein from C. elegans. And at the end of all of this, we end up typically with around 200 nanograms of the native BLTP1 protein complex, which is sufficient for cryo-EM studies, but is not sufficient for the majority of biochemical studies. For example, like running just an SDS page gel, you wouldn't be able to see the protein with small amounts of protein. So the first thing that we did, though, when we had sort of figured out our entire purification strategy from worms, was not cryo-EM, but mass spectrometry, because we wanted to know, are there any proteins that are co-purifying with BLTP1? Is anything coming along with LPD-3 that we should know about? So here we have the gene names in the left column, and in the middle, the peptide spectral counts for our transgenic worm line, where we were isolating LPD-3. And on the right, our control, which were wild type worms. So this was meant to be a control, like, is anything sticking non-specifically to the beads or whatnot, or other aspects of the purification process. And we saw that, yes, that LPD-3 was co-purifying with these two auxiliary proteins, which didn't actually have a gene name. They hadn't been studied in any organism. They had a very high peptide spectral counts and no peptide spectral counts in the wild type control. And so we ended up calling them LTAP1 and LTAP2, LTAP being lipid transfer auxiliary protein. And we refer to this one, though, LTAP1, primarily as spigot. And we haven't given LTAP2 a more interesting name at this point, though, because it's been a bit harder to characterize, which I'll show you in a bit here. So then we use this same purification strategy to do cryo-EM experiments. And we ended up with a really nice 2.6 angstrom cryo-EM map of the LPD-3 complex. So in this map that you see here, different proteins are shown in different colors. We have LPD-3 in green, the auxiliary proteins in orange and in yellow. Lipids, you'll be able to see some lipids in gray, and this transparent blob is the detergent micelle that's surrounding the transmembrane region where it inserts into the endoplasmic reticulum. So using this map, we were able to build a model of the protein complex, and we were really excited to see that the LPD-3 lipid transfer tunnel was just packed with lipid molecules. The whole space was filled with density corresponding to liquids. And so this yellow protein here, we determined as spigot. The orange protein is TMEM170, and its identity and function was actually determined by the Vani and Conibear groups because, as I'll show later, this protein didn't actually appear in our mass spectrometry data. So the three main takeaways from this complex is one, this is only half of LPD-3. If you were looking at the tunnel and thinking that it looked kind of short relative to those AlphaFold predicted structures I shown earlier, you'd be right. It extends to about double this length. And we think the reason that we don't get a high resolution map for the C-terminal half of the protein is probably some type of flexibility going on there that's preventing high resolution structure determination, but we're not entirely sure. We're still working on it. We also saw that the tunnel is packed with lipid molecules, which gives us some really interesting insights into how lipids are being transferred. And we saw that LPD-3 is bound to a few different auxiliary proteins. So first, looking at LPD-3, if we look at it and color each of these RBG domains in a different color, you can see how they're stacked in tandem in order to form the tunnel. So an RBG domain stands for repeating beta groove domain. And these repeating beta groove domains were sort of a known structural element of BLTPs and sort of a defining structural element of BLTPs. But it was really exciting to see them in this structure and to see how they're organized. So not only are they sort of stacked in tandem to form a modular unit, and the more RBG domains you have, the longer the BLTP protein tunnel, but also each RBG domain is rotated a little bit relative to the previous one, which gives the tunnel this sort of spiraling architecture, which you can see if you look at the beta strand backbone here. And if we look more closely at one RBG domain, you can see that it's formed by this beta taco here, so this V of the beta strands, and over the top has what we call a loop cap, which is typically comprised of one or more alpha helices along with an unstructured loop that sort of covers the top of the beta taco. And on the inside, the amino acid composition is sort of like really ideally suited for solubilizing lipid molecules. Most of the amino acids are hydrophobic, as one might expect for a hydrophobic lipid. But then what we also saw are these charged residues that line one side of the tunnel, and they sort of continuously line the right-hand side of the beta taco all the way through the tunnel. And as we'll see later, these charged residues interact with the charged head group of the lipid molecule in order to organize all of these lipids within the tunnel. And the RBG domains also varied substantially in length. So the first few RBG domains were pretty short, and they got longer and longer as it progressed towards the midpoint of the tunnel. And also they got flatter as it progressed towards the midpoint of the tunnel, and they opened up more, allowing more space for more lipids. We saw 26 lipid molecules within the inside of the tunnel. And if we look at this image here, the structure where it's sort of been sliced in half in order to see the lipids on the inside, you can see that it's packed with lipid molecules, and also the diameter of the tunnel changes substantially as you go from the beginning to the midpoint. So in the beginning, it's relatively narrow and only fits about one row of lipids. And then as you get towards the midpoint, it sort of opens up substantially, allowing up to 5 rows of lipids sort of stacked all together. And if we zoom in here, you can see each of these rows is colored gray or gold, and they're all stacked, one right on top of each other. And the density of these lipids within the tunnel, interestingly, is about the same density of lipids that is observed within a membrane leaflet. So it's like a little chunk of membrane leaflet within the tunnel. And then again, if we look at one specific lipid, you can see how it's organized by a combination of these hydrophobic residues shown in gray and these ionizable residues, these charged residues that are shown in red and in blue, where these red and blue residues are interacting with the head group of the lipid molecule, which is charged. And then the acyl tails are sort of nestled in that hydrophobic V of the RBG domain. And this here is the very first row of lipids in the tunnel, the one shown in gold here. And you can see essentially what we saw in the cryo-EM map, the density, which is shown here in mesh. So in some cases, we only saw a partial lipid tail, and we didn't see the rest, presumably due to flexibility. And in some cases, we saw the entire tail for the whole molecule. But importantly, we saw primarily like little V-shaped structures within the tunnel. So therefore, we weren't able to determine the identity of these lipid molecules. So they were all modeled as phosphoethanolamine because BLTP proteins are known to transport phosphoethanolamine. We also saw openings in the side of the tunnel along the entire length of the tunnel. We call these openings hydration portals because it looked to us like these openings were very intentionally sort of structurally placed in order to allow water molecules from the cytosol to enter and hydrate the lipid head groups. So I sort of naively thought going into this project that these tunnels would be sealed tubes traversing the cytosol, which was not at all true. And you can see all of these hydration portals and the little head groups of the lipid molecules shown in red that are peeking out from inside. And again, we think that these openings exist in order to allow water molecules to enter and hydrate the charged head groups of the lipid molecules in order to facilitate flow, possibly through the tunnel. And these hydration portals are especially apparent if you look at the tunnel sort of as slices, like you would slice a hot dog. And in some regions, it's completely closed, like here. But then in other regions, there's a clear opening, and that opening is positioned directly above the head groups of the lipid molecules. So again, just looking like water is entering the tunnel. And before we observed that the packing density of the lipids towards the midpoint of the tunnel was about the same as that is in a membrane leaflet. And here, the fact that we have water molecules also entering and hydrating the lipid head groups makes it seem even more like a little piece of a membrane leaflet that's moving through the tunnel because this little region of membrane would be experiencing sort of the same environment that it would be experiencing in the cytosol, where its head groups would be hydrated and its tails, the acyl tails, would be within a hydrophobic environment. And then looking at the auxiliary proteins, so we have here within the cryo-EM structure, two auxiliary proteins that we observed. So spigot, shown in yellow, TMEM170, shown in orange. But then in our mass spectrometry data, we saw not TMEM170, but we actually saw a different protein that we labeled LTAP2. And we did not see LTAP2 in our cryo-EM map. We think probably because LTAP2 is localized somewhere at the C terminus of the protein. And we didn't see the C terminus at high resolution. And TMEM170 we didn't see because it's localized almost entirely within the membrane. It's composed of 3 transmembrane helices. And the way that we do mass spectrometry, transmembrane proteins don't tend to show up as strongly within the mass spectrometry data. So knowing this about what auxiliary proteins are present, our two main questions were, are these conserved? So here we're looking at the C. elegans complex, and are these auxiliary proteins also found in other organisms associated with BLTP1 proteins? And two, what are they doing? Like, what's their role in lipid transport? So we set out to answer these questions by working with Yoon-Sik Kang, who's going to be talking next about his work on BLTP1. And he essentially knocked down each of these proteins in a variety of different organisms and then assessed whether that knockdown or sometimes knockout phenotype mirrors the knockdown phenotype of BLTP1, because that would tell us, are these proteins well, it would give us a lot of information about whether these proteins are working with BLTP1 in that particular organism. So I'm not going to go in detail into the experiments that were performed, but with spigot, we saw that the knockdown phenotype in C. elegans, in flies, and in HeLa cells here, we're actually here looking at co-localization, But all of this information suggested that spigot is in fact part of BLTP1 complexes, and so is a conserved member of BLTP1 complexes across species. But then what is its role in lipid transport? Looking at the cryo-EM structure, it's not totally clear to us. So it's obvious that spigot is really important for BLTP1 simply because it interacts really extensively with BLTP1. It interacts with the transmembrane domain and then sort of winds its way around the outside surface of the tunnel, ending in these 4 helices, which are embedded in the side of the tunnel. So these is very extensive interactions, but what its actual role is, whether that's stability, localization, regulation of liquid transport, isn't clear at this point. And then for LTAP2, its role is even less clear. So UNSIC did a similar set of experiments, knocking down LTAP2 in worms and in flies, and the results were a bit ambiguous. So at this point, we're not really sure whether it's a conserved member of BLTP1 complexes. And its role in lipid transport is not clear because we were not able to observe it in our cryo-EM map. So LTAP2 is very much still under investigation. And then for TMEM170, so TMEM170 was investigated by the Conibear and Vani groups, and they showed that TMEM170 is not only a conserved member of BLTP1 complexes, but they showed that its role in lipid transport is as a scramblase. So in this, they used molecular dynamic simulations and showed that there's a lipid scrambling pathway that leads up the center of these 3 transmembrane helices here, which is shown in gray. And looking at TMEM170 within the cryo-EM structure, and if we map that lipid scrambling pathway, it sort of leads right to the edge of the BLTP1 tunnel. So the idea is it's scrambling lipids between those 2 membrane leaflets and could potentially be part of what is feeding lipids into the BLTP1 tunnel based on its position. But at this point, we don't know for sure. But so what about all of these other proteins? So in our mass spec data, we definitively identified LTAP2 and spigot based on their very high peptide spectral counts as members of the BLTP1 complex. But then we saw all of these other proteins, which are present in much lower abundance, but perhaps also play a role as transient interactors. So UNSIC did a phagocytosis assay with these proteins to assess maybe they are important for BLTP1 function. And the answer for most of them was yes. So it's possible that these proteins are also playing a role in BLTP1 mediated lipid transport, possibly as regulatory proteins, adapter proteins, things like that. So in summary, the main things that we learned from our structural and cell biology experiments of BLTP1 were that first, within the tunnel, the phospholipids are organized via this track of ionizable residues, which in our little cartoon here on the left is shown by this tie-dye arrow. So we have that string of charged residues that together are organizing all of the charged lipid head groups so that they're all pointing the same direction and they're all organized in the same manner within the tunnel. And second, we saw these hydration portals, these openings within the tunnel that we think allow water molecules to enter and hydrate those lipid head groups, probably facilitating the diffusion of lipid molecules through the tunnel. And then we observed that spigot and maybe LTAP2 are conserved members of BLTP1 complexes, as well as perhaps TMEM170. And so finally, just like to acknowledge everyone in my group, which is a fantastic group of people, especially Hannah, who did a lot of the C. elegans work with BLTP1. We do all of our cryo-EM at PNCC in Portland, and these are our funding sources. So thank you for your attention. I'd be happy to take any questions.
All right, thank you so much. That was fantastic. We're going to open it up for questions. And so the first one I see, okay, I'm going to allow it to talk. Uh, uh, is it Binks Waterberg? Yeah, you can
It
unmute
is.
yourself and ask. Okay.
Yeah, this is Bing Swattenberg. Wow, that was so cool. Um, I'm really struck by the organization of the lipids into what you describe as being sort of leaflet-like organization, sort of towards as they progress along the tunnel. And I'm wondering what you think about whether that may drive some directionality of the lipids, since they would be sort of self-organizing and less monomeric, and sort of opposing the scramblase activity at the N-terminus.
Yeah, I'm not sure how the organization may impact the directionality, but the width of the tunnel is something that I wonder if it impacts the directionality, how it's much thinner at the entrance, at the end terminus, and then it widens substantially towards the midpoint. And I wonder if that somehow could be impacting directionality and flow. But the organization, I don't know.
Yeah,
Right. Oh, oh, oops. I'm sorry. I mean, uh, that's my bad. Sorry, thanks. Technical difficulties on my part. Sorry, we're gonna please put your other questions in the chat as well. I'm gonna move on to George if he has some question. You can unmute yourself and ask.
Thank you, Sarah, for the beautiful talk. Very interesting. I was wondering, like, so you have these portals which allow water to enter into the channel, into the tunnel. Uh, in the absence of lipids, wouldn't that destabilize the structure? And do you think that this may potentially explain? Do you always need lipids in order for it to fold? And could this explain why these proteins are maybe difficult to overexpress?
Sort of yes to all. So I do think that the tunnel is sort of always filled with lipids. And I wonder if even as it folds, as it's translated and folding, if it folds in the presence of lipids within the tunnel. So my guess is that yes, the tunnel is sort of always packed with lipids because otherwise it might be structurally unstable. And so that could be definitely a reason why it's difficult to recombinantly express is because it has unique folding and assembly requirements in that way.
Okay, thank you.
Mhm.
Okay, one super fast question, Monica. Can go.
Hi, thank you. This is a fantastic work. I'm curious about your mass spec data. So
Mhm.
I saw the LDH having like an important role there. Do you have any idea of what this metabolic enzyme is doing there?
No, sort of at this point, this is honestly kind of all the data that we have that these proteins, when knocked down, have a phagocytosis defect in flies. But whether or not that phagocytosis defect is even related to BLTP1 function or just, something within the broader scheme of phagocytosis is kind of unclear. This was a broad strokes initial assay.
Okay, perfect. Thank you for the questions. Okay, now we're gonna move on here. And so it's really my pleasure to introduce our next speaker, Dr. Yunsik Kang. So Yunsik received his PhD, like me, at the University of Wisconsin-Madison. He did his postdoc work at the University or Oregon Health and Science University. He started his independent lab at the University of Colorado School of Medicine last fall in the Department of Cellular and Developmental Biology. So I'm super excited to hear your presentation, Yunsik, on the function of BLTP1, kind of at a different scale. So take it away.
Okay, can you all hear me?
Yes.
Okay. Thank you, Michael, for that wonderful introduction. Like, I would like to say thank you, Sarah, for that wonderful talk. If you think of Sarah's talk as like the main feature movie, mine is like the prequel. So this is where we kind of go back in time and figure out where we started with BLTPs and how we got interested in the transfer proteins. So my lab in general is not interested directly in BLTPs, but in the process known as neuronal remodeling. And this is when, during development, as you can see in this classical experiment, where the brain is initially overly producing neurons. So they are massively overwired and overproducing neurons. And what happens during development is you have to eliminate certain neurons and connections to form the final mature circuit. Now, this is not only important to build the proper nervous system, defects has been connected to neurodevelopmental disorders such as autism and schizophrenia. And this occurs through two mechanisms. One is known as local pruning, where you lose certain processes, or cell death, where entire cells are eliminated. And a lot of work from many labs have highlighted that not only neurons are important, but the glial cells, which are half of your brain, play a critical role in this process of neuronal remodeling. So in my lab, we have three fundamental questions that we want to really answer. It's number one, how do the neurons make this decision? It's clear that they are the decision makers. They use some molecular mechanisms to decide what they're going to eliminate. And how do the glial cells participate in this process? Basically, how do they come and communicate with the neurons to execute this remodeling? And finally, in a lot of places, in some instances, 50% of neurons die within 2 days in development. And that will lead to a lot of neuronal debris. So we need to clear all that debris, and the glial cells are the main cells that do this. So how do the glial cells recognize, engulf, digest, and clear all that neuronal debris? And to really focus on today's talk, I'm going to really focus on this third question of how glial cells phagocytose neuronal debris. And to understand these three fundamental questions, I think you need a system in which you have of course, neuronal remodeling occurring. You need neurons and glia. And you also have to there has to be a very strong genetic organism to do molecular analysis. And I think the perfect system for this is the fly brain. So here we're looking at a fly brain. These are the two brain lobes. And this is what is known as a ventral nerve cord, which is function analogous to mammalian spinal cords. And this is how the brain is laid out. We have what we know as a cell cortex, where the neuronal cell bodies are, or somas, are present. And then they send their processes into the white region, known as the neuropil, where all of the synapses reside, and most of axon and dendritic processes are present. The flies are a unique system because this brain development is very stereotypical, meaning that we can label a subset of neurons over and over again and ask a simple question: how do they remodel? Not only can we do that, we can label a single neuron and ask how this single neuron goes through remodel. And the main glia cell that I'm going to focus on today are the astrocytes. Now, I don't lightly call these astrocytes. There's a lot of work from many labs showing you that these are bona fide astrocytes that are analogously similar to the functions of mammalian astrocytes. And the final question is, does fly actually do remodeling? And the answer is yes. And the stage that I focus on is known as a metamorphosis, where a crawling larvae transfer to a mature adult. And within 4 days, what happens is this larval nervous system has to be broken down within the first 24 hours and get reorganized to make a more complex behaving adult fly. And this is a unique system in which we can understand how remodeling is happening because, again, the breakdown is very stereotypical, just like the brain was initially built. So let me show you the extent of how much remodeling is actually happening. So here on the top right panel, I will always show you an image of the entire nervous system and a red box of where I'm imaging. These time points are critical to understand the talk. So larva is like before metamorphosis. So you can think of it as the default. And hours indicates hours into metamorphosis. So in this case, we're looking at an entire nervous system. And this is a maximum Z projection, looking at the entire nervous system. And we're labeling a presynaptic protein known as Brook pillow. So this is not a region of the fly brain. This is the entire fly brain looking at all of the synapses. And you can see within 12 hours, most of the synapses are eliminated. And we know that this is not GFP-demic. This is true synaptic elimination. Because 48 hours into metamorphosis, if we take an EM image, we cannot find a single synapse. And we know the astrocytes are the main glia cells doing this because if we knock down a single phagos receptor known as Draper, which homolog MEGF10 has been shown to play an important role in astrocyte-mediated synaptic pruning, you can see we have a really strong retention of the synapses, showing you that the astrocytes are the main glia cells clearing all this synaptic debris. So how do the astrocytes perform phagocytosis? Now we're looking at the astrocytic membrane here in green and the synapses again in magenta. And the cell bodies are outside of the neuropil, and the astrocytes send their processes to closely associate with the synapses. Now, these are the astrocytes that most neuroscientists study, the ones that are closely associated, clear neurotransmitters, make sure that the nervous system runs properly. But within 2 hours into metamorphosis, every single astrocyte in the fly goes through a dramatic transformation. You can see that now the processes start forming these circular structures. And if you look later on, they start fusing and making these larger clustered structures. And inside of these vesicles, you can see synaptic material. Not only that, we can see neuronal material, and at the EM level, we can see that they're packed full of neuronal debris. Show you that every single astrocyte in the fly, within the first couple of hours of metamorphosis, all become phagocytic. For a lot of work, we really now have a really good understanding of the cell biology of how these astrocytes perform phagocytosis. They make early phagosomes, fuse, make larger late phagosomes, and eventually make these larger clustered phagolysosomes to clear and digest all the debris. Furthermore, we did a lot of tests of known molecules that play a role in macrophage phagocytosis. And then we looked and found that they do the same thing in this context. So this is a highly conserved mechanism that is executing, we're using this known molecular mechanism to execute phagocytosis. So some of you might be thinking, this is great, but I can do this in macrophage cell line. What's the power of this doing in the fly? And let me tell you what you can do in the fly. Is that when you have a really good understanding of cell biology of how this can happen, you can do an in vivo genetic screen to find new molecules that play a role in astrocyte phagocytosis. So let me walk you through how I did this screen. So rather than looking at all of the synapses, I decided to look at one neuron that we know that goes through remodeling, known as the corazona neurons. You don't have to know what these neurons are, but the most important thing is these neurons go through canonical apoptosis or cell death. And then you can see they get fragmented and eventually get cleared. And the clearance, of course, is happening because the glial cells are phagocytosing all of this neuronal debris. So you can knock down a single gene that is critical for phagocytosis, and that should lead to a retention of this neuronal debris. So this is important that we are imaging the neurons, but the knockdown is happening in the glial cells. So what we decided to do is to do this to all the transmembrane proteins in flies and all the secreted molecules in flies, which is about 1,333 genes, knock them down one by one, and then image them 12 hours into metamorphosis and asked, do they have a phagocytosis defect? When we did this, we got 94 hits. And today, because of the theme and what I got interested in, and I'm going to talk to you about what we did and what we try to understand with this molecule called tweak. The reason I picked tweak was it was the most consistent and it get a strongest phenotype, one of the strongest phenotypes in this screen. And it was a lot of parameters it passed through. So we decided to study this molecule. This molecule, we wanted to make sure that this molecule is playing a role in the astrocyte. So we did our genetic homework. Again, these are the corazon neurons getting completely cleared. If you knock down tweak in only the astrocytes, you get, again, neuronal debris. When we look at a loss of function mutants, these mutants are extremely sick. Very few come out of embryogenesis, but if you do a lot of work, you can get some of them. And you can see at the null mutants, you get, again, a loss of function, a null mutant over like a complete deletion of the locus, which is probably the strongest loss of function you can make, results in defects in neuronal debris clearance. To confirm this, what we did is we expressed tweak back into the astrocytes in the mutant background, and we can completely rescue, showing that the astrocytes are the critical cells that are using tweak to perform phagocytosis. Now, when I found tweak back in the day, it was all I knew about it. It was a highly conserved. It was a massive protein. It's the 33rd largest protein in flies. It's 565 kilodaltons. And there's only one paper about it, two papers, but it was mostly from Hugo Bellin's lab, showing that it played a role in synaptic vesicle endocytosis. I also was really interested in this molecule because it had a strong association with a neurological disease known as cryo-electron syndrome. The patients had severe brain malformations, and they had ASD-like behavior. So with this knowledge, what I decided to really study what this molecule was doing. And to be honest with you, for many years, I didn't know what it was doing. But until AlphaFold came out and many things happened, and now you will know, listening to Sarah's talk and what Michael just told you, The tweak is BLTP1 in mammals, it's LPD-3 in C. elegans, and it's a bridge-like lipid transfer protein. Here's the alpha-fold predicted structure of the entire tweak protein. Here's a single transmembrane domain, which will be embedded in the ER, like Sarah said, and you can see it makes this large tube-like shape with a hydrophobic cavity inside. So now if you think of the cell biology and the in vivo function of tweak, well, does tweak play a role in phagocytosis? And what is the literature showing? There was not a lot of literature. There were just some screens that were done. And what was interesting is this screen done in C. elegans showed that LPD-3 in an RNAi screen in a single chromosome had a really strong defect in apoptotic debris clearance. At the time, as Michael said, there was known as KIAA1109. This is a tweak homolog. Seemed to be important in a screen that used magnetic beads. It was found to only have a defect in engulfing large magnetic beads. But this paper really came out in the Bessic lab, which was really exciting, that showed that there's this particular bacterium that enters a cell using a phagocytosis machinery, and they survive and they will replicate and eventually burst the cells to spread. And what they found when they did a screen for surviving macrophages after this bacteria infection, that KIA1109 or BLTP1 or human tweak was critical for this bacteria to enter and kill the cell. This, in hindsight, is amazing because this chromosome 1 open reading frame 43 that pulled down as the number, only the second hit that blocks the same pathway as tweak, is actually the homolog of spigot, the molecule that Sarah just told you about, showing you that this complex, perhaps not only just in flies, throughout evolution might be playing a role in executing proper phagocytosis. So the first question I want to ask is, where is tweak expressed? To answer this question, I used a nuclear reporter. So what this means is when tweak is transcriptionally turned on, that cell's nucleus will light up. So on top here in cyan, we're looking at all the nuclei, and this yellow region here depicts and outlines where the neutrophil region is. So everything inside these cell bodies are all astrocytic cell bodies. And as you can see in the tweak nuclear reporter, as metamorphosis goes on at larva stages, you don't see much expression. But by 2 hours into metamorphosis, you start seeing only the astrocytes are turning on tweak. And then 6 hours into metamorphosis, it's really highly expressed. We wanted to confirm this nuclear reporter, so we use in situ hybridization. Again, here in green, we're showing the cell bodies of the astrocytes. At larval stages, very little mRNA, but by 2 hours, you can see a robust increase of tweak in both in the astrocytes, showing that tweak is expressed in the right time in the right tissue. So what happens when you get rid of tweak? Well, to answer this question and really quantify this transformation, we used a system in flies known as markup. So what this means is we can generate a mutant clone of astrocytes, a single astrocyte mutant clone in a wild-type, relatively well-type animal background, and label that mutant clone. So this is a control. As you can see, this is a cell body of the astrocytes, their processes. 2 hours into metamorphosis, they're starting to form these circular phagosomes like I just showed you. When you knock down tweak, you can see that there is very little phagosome formation, meaning that when you do not have tweak, phagosomes cannot properly form. We know that this is due to tweak because if we supply tweak back to these astrocytes, we can start making these phagosomes again. So here's a quantification. What I quantified is a vesicle number or phagosome number per astrocyte. And you can see at larva stages, I don't get any vesicles. But there's a significant increase of these phagosomes. When we make a tweak mutant clone, you see there's very significantly less. When you supply tweak back to these astrocytes, you can rescue the phenotype. And even if you supply BLTP1 or the human version of tweak back, you can also get a significant rescue, showing you the conservation of function. So how does tweak perform this function during astrocyte phagocytosis? To answer this question, I want to see where tweak localizes. So what we did is we generated an endogenously tagged tweak protein. Now, tweak in flies, like I said before, is lethal. Most of them cannot survive embryogenesis. But this endogenous tag was fully functional. Every fly survived There's no different from wild type and did not have any phagocytosis function, showing that this is a functioning protein. So here, what we're looking at in the top panel is the GABA transporter. So this is actually looking at the astrocytic membrane. Here in magenta, we're looking at endogenous tweak expression. Again, at larval stages, we see very little protein. But as they enter metamorphosis, the astrocytes are now turning on endogenous tweak. And you can see this robust increase 6 hours into metamorphosis. When we really zoom in and we can find a phagosome, for example, right here, you can see tweak is beautifully localized around this phagosome. And when you look at the periphery of the cell bodies, tweak seems to be around the periphery of the cell bodies. So with all this knowledge and what Sarah just showed you and of what we know about BLTPs and how they sit at ER contact sites to do lipid transfer, and one of the things they are known to do at these sites is to do lipid transfer to do membrane expansion. So for example, ATG2 or BLTP will sit at the ER isolation membrane to transfer lipids to build the autophagosome membrane. So because tweak is at the periphery of the cell and it plays a role in phagocytosis, my hypothesis was that during astrocyte phagocytosis, what might be happening is tweak is at the ER plasma membrane contact sites, transfer lipids to either supply lipids to build the help build the phagosome membrane or directly supply lipids to build the phagosome membrane. And if you don't have tweak, this is why you have a complete breaking of building this phagosome membrane because you just don't have the lipid supply. The next one I wanted to really look at was the ERPN contact sites themselves. We can do this by looking at a constitutively active stim construct, which has been shown in flies to really label the ERPM contact sites nicely. So at the larval stages, you can see these ERPM contact sites are throughout the neuropil. And this is really specifically expressed only in the astrocytes. So these are astrocyte-specific ERPM contact sites. And 2 hours into metamorphosis, you can see similar ERPM contact sites numbers. At larval stages, we see a significant decrease of ERPM contact sites numbers. And by 2 hours into metamorphosis, we start seeing these clumping of ERPM contact sites. And when you look at the cell bodies and we can kind of zoom in, what we found is that there were these large clumps of ERPM contact sites when we got rid of tweak. Now, we don't know if this is directly disrupting the ERPM contact sites because when I do an ER marker, I see the same kind of clumping. So this could just be ER morphological defects. But currently what we're doing is we're doing a lot of EM to really show when you lose tweak, what are these structures of this clumped ER to try to understand what happens when you lose tweak. So overall, I think this really shows you that tweak is perhaps an ERPN contact site protein that plays a role in transferring lipids during phagocytosis to supply the lipid chain for efficient phagocytosis. And finally, what I really wanted to do was kind of go after this disease, the unclear syndrome mutations. And the way I did this is I specifically focused on this one mutation, this asparagine to lysine change, because this asparagine is conserved throughout evolution. So using CRISPR-Cas9 genome engineering, I generated a asparagine to lysine mutation. This means in the whole fly genome, there's only a single missense mutation, that's this asparagine to a lysine. And I asked, what is the phenotype? And what I was really surprised to find is that this is one of the strongest loss of functions that I can find. It replicates everything from a complete deletion. It has a strong phagocytosis defect. It does not survive past embryogenesis. It has a significant disruption similar to what I just showed you for ERPM contact sites. And these are the kind of things that I want to do in my lab where we're trying to use these known disease mutations to not only understand the etiology of the disease, but using these mutations to try to get at the molecular nature of how these proteins are functioning. And also, just like Sarah just stated, this IPMS spec for me is like a gold mine because we can start really understanding how these function during the process of astrocyte phagocytosis. And most of these have an astrocyte phagocytosis defect. And I've done many, many screens. This is a high percentage of getting hits. Usually you get less than 10% if you're lucky. Sometimes you get less than 1% when you do these screens. So this is a really high number. And currently, I'm really focusing on the top two interacting proteins. And I was very happy because recently we generated a spigot null mutation in flies and an LTAP2 null mutation in flies, and they're all lethal, showing that perhaps they're all functioning similar to tweak, where the flies cannot survive and perhaps playing a role in phagocytosis. And in the future, I want to also start looking at these transient proteins to see what they might be doing in the context of lipid transfer, but also at the same time, phagocytosis. And I want to really kind of step back and talk about my lab a little bit, because this means that this is 1 out of the 94 that I find. And if you think of this, this is amazing. And there's a lot of other molecules that our lab is really interested in following up and understanding how astrocytes are performing this amazing feat of phagocytosis. And finally, this is, again, one of the 3 questions my lab really wants to answer. We really want to understand how neurons and glia interact during phagocytosis, but also during development and neuronal remodeling. So with that, I would like to end my talk by saying that hopefully I've shown you that fly metamorphosis is a powerful system to understand neuronal remodeling. We have developed many tools to define different stages and molecular features of astrocyte phagocytosis. And I want to end by saying that although genetic screens are old school, and I believe they're classics, but people think they're pretty old school, but I think it still has a place in science, because you can find these really interesting new biology. For example, how these BLTPs might be playing a role in the process of phagocytosis. And I think, this is like me doing this talk is a representation of that, because I never would imagine I would be giving a talk at a lipid seminar. So with that, I would like to thank my lab. I have 1 member right now, but hopefully in the summer, it's going to grow up to 7. I want to thank my funding sources. I also want to thank Sarah. Sarah really showed me the power of a protein purification and introduced me to the beauty of structural biology. I also want to thank John Peters from University of Richmond, who does a lot of MD simulations, and we have a collaboration going. A lot of the work that I did today was done when I was a postdoc in Mark Freeman's lab at the Volume Institute, so I want to thank them. Of course, my funding sources are my postdoc and my funding sources currently, and I'll be happy to take any questions.
All right, fantastic, Insek. Thank you so much. We're going to open it up for questions, and I see one right now. So go ahead, George.
Thank you for the talk. It was super interesting, and it was nice to have them back to back like this. So I was just thinking about the question I asked to Sarah about the role of lipids in the biogenesis of these lipid transfer proteins. And based on your work, it suggests that they may be localizing to, because at ER-PN contact sites, you also have lipid transfer. So could it be you have association with sites of lipid synthesis? And thinking back to Sarah's table of hits, I saw fatty acyl synthase, FASN was a hit on there as well. So just a comment / question. And I have another question, a proper question, but I can stop here and allow for others and come back to the other one later.
Yeah, so thank you for the question. You're absolutely correct. Like, I think one of the main questions in the BLTP field is MD simulations don't really show that these can quote unquote select for lipids, but it could just be literally like the ER is generating specific lipids. And it makes sense to me that BLTP1 or BLTPs have to be at where lipids are being synthesized. And of course, like you said, this protein FASN1 coming up is very exciting, but as you can see, it didn't have a phagocytosis defect. I think that could be because there's in flies 2 of them, there's 1 and 2. So I have to probably knock them both down, but that's an absolutely fascinating question that we hope in the future that we can follow up on.
Okay,
All right.
Thank you.
Yeah, thank you so much for your question. I'll go on to the next question. Unmuting now. Fikadu, please ask your question.
Yes. Hi, that was a very nice talk. I was very surprised to see that kind of really transformation from one state to, phagocytes. Have you done general lipidomics during those transformations of the astrocytes?
You know, I haven't, but now listening to you, I think I should do that. That's a really good idea. Yeah, that would be fascinating. Yeah. Wow. I should write that down and do that experiment. Yeah, that's a great idea. I have not done it, right? I've looked
Yeah,
at
I mean,
different
it's the,
lipid.
yeah,
Yeah.
I think the same goes to in the presence and the absence of tweak, right? In the
Yes,
wild
yes,
type
yes.
and the mutant. I think it would be an interesting one.
Yes. I think the only problem with that is like I don't know how scalable it is and how I can just get the astrocyte out. My understanding is like sorting them will kind of disrupt the lipids. So I think there's more thinking about what we have to do, but that's actually a really interesting and great idea. Yeah.
Last question here, Samshad, you can unmute yourself.
Yeah, that was a very nice talk. The other cell type in the brain is microglia, which are the professional phagocytes. What happens in a fly? Do you also have microglia, and do they use tweak as well?
Yeah. So in flies, we don't have microglia. So the main cell that does all this work are the astrocytes. But what's really interesting to me is that we have done in flies, like if you think of maybe a microglia-like situation, it's like injury, right? So what we can do in a fly is we can rip their antenna out, and that will, of course, lead to exon degeneration. And then all of that debris has to be cleared by glia cell type in flies and sheathing glia. These glia cells react like microglia. They're usually very like not doing much, but then once you have an injury, they kind of become extremely phagocytic, send their processes in and just gobble stuff up. What's interesting is when we knock down tweak in the sheathing glia or in glia cells and do an injury, they seem to be not playing a role. So it almost seems like tweak is transcriptionally turned on during metamorphosis. Is almost like getting ready. They know there's a development program to say, let's turn on these BLTPs to start phagocytosis. But when you have an acute injury, there's no response from tweak, and that's not what the mechanism is. So which I find interesting, and that's why that screen of the magnetic beads where tweak plays a role in larger magnetic beads, but not smaller ones, could be because maybe when you have a more stronger demand to make more membrane is when tweak or BLTPs are required during phagocytosis.
Very interesting. Thank you.
Yep.
All right, so we are at time. Now please help me thank our two fantastic speakers, Sarah and Yunsik. Thank you both for those outstanding talks. And thank you all for attending. I really appreciate it with all your questions and engagement, and I hope to see you next time.